Hello everyone, my name is Arpita Das and I have brought to you the concept of Operon or gene clusters. To begin with, I would like to say that genes and cistrons, these two terms are used synonymously. When we say gene clusters, we can also say polycistronic units. So our operons are basically polycistronic units. Where are they found? Mostly in prokaryotes, because when we talk about eukaryotes, they like to keep monocistronic units. That means one promoter for each gene. So it's basically found in prokaryotes. There are exceptions though. How was it discovered? Discovered by Jacob and Monod by conducting a series of experiments on E. coli mutants, to be specific, and as a result they came across this Lac Operon model, which is extensively studied, and we will be using this as a reference to begin with to understand our operon. What are the types of operons? Two types, catabolic and anabolic. The Lac Operon is catabolic. As we can see in the diagram, there are three structural elements, Z, Y and A. Z codes for beta galactosidase. Y codes for permease, which is a channel protein that helps in the uptake of lactose, and Lac A for alpha transacetylase. We also have cis elements present, which are our P-O promoter operator, our CBS or the cap binding site. These are called the cis elements since they are present in the main DNA sequence. And these Lac Z, Y, A are called the structural elements. This unit of Lac I and O works independently or functions independently to produce a repressor or an inhibitor which can regulate the switching on or the off of the Lac Operon. Coming back to the types of the catabolic and the anabolic operons, example of an anabolic operon will be the tryptophan operon. Catabolic meaning catabolism. So in this case we will be looking at the lactose catabolism or degradation, degraded into the glucose and the galactose. Your anabolic or the tryptophan operon model will focus on the study of tryptophan production. So we are looking at Lac Operon. Simple explanation would be when lactose is present, the operon is switched on. When lactose is absent, it is switched off. So this is an example of positive gene regulation. Negative gene regulation would just be the opposite, right? Let's first take into consideration the case where lactose is absent or very much low in concentration. So what happens? There's no lactose. So this Lac I gene will be encoding for a particular protein which has the capability to bind to the operator. When it binds to the operator, the RNA polymerase cannot come and bind to the promoter, and as a result the transcription will not occur. So in this case our machinery is switched off. When we have high lactose, however, lactose gets converted into allolactose, which acts as an effector molecule for this particular protein transcribed by Lac I. This now, since it has undergone conformational change, cannot bind to the operator sitting here, which makes RNA polymerase come and bind with the promoter, and hence our transcription becomes possible. In this case our Lac Operon is switched on. Now the Lac Operon by default remains switched off. Now let's look at the effect of glucose on the upregulation or downregulation of the gene products. When we have low glucose concentration, a particular product called CAMP, which is synthesized from ATP by the use of adenylate cyclase, which is an enzyme, it binds to the cap, which is a CAMP binding protein. This CAMP and cap unit further binds to a CBS, which is the cap binding sequence or the site which is present in the main chain of the DNA. This binding upregulates or increases the affinity of the promoter to the RNA polymerase. As a result, the production of the gene products will be maximized. When we have high glucose concentration, however, the conversion step of the ATP to the CAMP is not possible, and hence a moderate expression is seen, because now there will be no boosting factor that was previously produced by the CAMP cap binding complex, right? Next up, so what are the conditions for maximal transcription of Lac Operon? As you can see written on the screen, low glucose and high lactose condition. That means, why do you need the Lac Operon in the first place? You need it because you need to convert the lactose into the 2 monosaccharide units, right? So if you have glucose as the primary source of energy, there's no need to break down the lactose, correct? So the glucose will be uptaken by the E. coli first, and then lactose will be broken down. So whenever there will be low glucose, only then, and high lactose, only then will you need your Lac Operon to function to degrade your lactose products, right? So that is the case that is shown in the diagram. So you can take it for reference. Now, who did the experiments? Jacob and Monod. They loved experiments and they conducted many experiments with E. coli mutants. We will look at the naming of the mutants and what phenotypes they create in the upcoming slide. However, I'd like to point to you 2 different terminologies, very important. Cis elements. Cis elements refers to the promoter, operator and the cap binding sites, which are a part of the main chain of the DNA, right? Now, the trans regulators are basically molecules that had been created by the cis elements itself in most of the cases. What these trans regulators do, as the name suggests, they regulate the expression of the gene, right? But then, how do they do it? They attach themselves to the cis elements and bring about allosteric regulation. Example, I can show you the cap or the CAMP binding protein that you had seen out here. This CAP had bound with the cyclic AMP, which in turn, that complex had bound to the CBS, which was close to the promoter, and as a result it had boosted the expression of the gene products, right? So now, done with the terminologies, we can go on to the mutants. Mutants would be constitutive mutants or OC, promoter mutant or P-, the beta galactosidase mutant, also known as the Z- mutant, the permease mutant or the Y- mutant, and the inhibitor mutants of 2 types, I- and IS. The I- mutant switches on the Lac Operon and the IS switches off the Lac Operon. Let's look at the mechanism of how these things are brought about and also the phenotype that will be produced. So the constitutive mutant is basically the case where your promoter, operator, Z, Y and A, this is the basic structure. This operator is not present. That means the control element is only not present. Since this is not present, the repressor will not come and bind out here. Since the repressor can't bind, this particular unit will always be on, or it will always keep the Lac Operon to be switched on. When we talk about the promoter mutant or P-, that means the promoter will be not in the position to bind to the CAP or the RNA polymerase, right? And as a result, the Lac Operon will be switched off. The beta galactosidase mutant will not lead to the production of the beta galactosidase. If that's not present, then lactose degradation is also not possible, and as a result, your glucose and the galactose will not be produced. Your permease mutant means the protein or the channel protein that was required to take up the lactose from the outside of the cell to the inside will not be present, and as a result, this transfer will not occur. So this will also decrease your gene expression. Inhibitor mutants are typically important. Why? Because out here the repressor, if you look at the structure of the repressor, it consists of two binding sites. One is the DNA binding site and the other is the allolactose binding site, which is the APO inducer. Okay. Now what happens? This DNA binding site, whenever it is mutated, it is the I- mutant. That means it will be absent. So if this is absent, this particular repressor cannot go and bind to your transcription unit, and as a result the operon is always switched on. Now when you have allolactose binding site missing, this will not lead to the conformational change, right? And as a result our Lac operon will always be switched off, right? So these were the phenotypes, and all the diagrams that has been used in this particular presentation is from bio.libertext.org. So a big thank you to them, and I hope you liked the video. It was a short one, so subscribe to my channel and stay tuned for questions on the Lac operon. Thank you.